The lens system continued. The ocular, or eyepiece, as it is more commonly known, is positioned at the top of the instrument. It consists of 2 or more internal lenses and usually has a magnification value of 10x. 3 or more objective lenses are usually present. They are attached to a rotating nose piece under the eyepiece. Each objective lens can be rotated into position over the slide. On a standard microscope, there are usually 3 objective lenses, each with its own strength of magnification. 10x, low power. 40x, high power. 100x, high power oil immersion. The third lens system is the condenser, which is located under the stage. Its function is to collect and direct the light from the lamp beneath it to the slide being examined above it. Introduction to phase contrast microscopy. A large range of biological specimens are virtually transparent when observed under a bright field microscope. In order to make the specimens easier to see under the bright field microscope, it is common practice to stain them. Staining, however, is not suitable for work involving living cells, as the staining process will kill them. To overcome this, a technique called phase contrast microscopy is used to make unstained objects yield a contrast image visible to the eye.